Welcome back to Enzyme Regulation, Control of Enzyme Activity. This time we will discuss allosteric effects and how these can alter enzyme activity. To get the most out of the videos, I recommend watching them in order. This is a reminder that this presentation is Dr. Johnson's intellectual property. Enzymes can bind ligands at a number of different locations. In terms of affecting the activity of an enzyme, a ligand can bind either at the active site or it can bind at a different site, called an allosteric site. Because only one molecule can occupy a given space at a time, a ligand other than the substrate that binds at the active site will be an inhibitor, usually competitive towards the substrate. Product inhibition is a good example of this type of inhibition. If the ligand binds at an allosteric site, it exerts its effect on the activity through long-range interactions. These effects may be either inhibitory or activating. Allosterically regulated enzymes may have one or more allosteric sites. The word allostery comes from the Greek allo for different and steri for structure. You will have encountered allostery when you studied hemoglobin. Although allostery was known from the early years of the 20th century, it was not recognized as applying to enzyme activity until the 1960s. Scientists working with allosteric enzymes prior to then noticed unusual kinetics that did not match Michaelis kinetics, but seemed to have little understanding of the reason for this kinetic behavior, which they very often described as complex or even peculiar. By the early 1960s, most of the major metabolic pathways had been worked out, and the connection between these pathways and the non-Michaelis behavior of the enzymes could be connected. The enzymes exhibiting non-Michaelis kinetics are situated at key steps in metabolic pathways. In many cases, the allosteric effectors are produced elsewhere in the pathway. These types of effectors may be either feedback inhibitors, produced further along the pathway, or feed-forward activators, produced earlier in the pathway or in a complementary pathway, and indicating an abundance of these metabolites and therefore of starting materials. The example pathway here is for isoleucine biosynthesis. The end product, L-isoleucine, is a feedback inhibitor for the first enzyme in the pathway, threonine deaminase. This is one of the first enzymes recognized to have allosteric regulation and the first metabolic pathway in which feedback inhibition was observed. While the classical models of feedback inhibition show the end product of the pathway inhibiting the first enzyme of the pathway, it is possible to have other products of the pathway inhibiting enzymes at other locations in the pathway. In allosteric regulation, the enzyme is sensitive to the concentration of the effector. The Kd for the ligand does not change. Therefore, if concentrations of the effector increase, a greater proportion of the enzyme will exist as an enzyme-effector complex. This means that the enzyme's activity will depend on both the effector concentration and the substrate concentration. We can see this relationship for both positive and negative effects in Michaelis plots. The enzyme activity curve clearly does not follow Michaelis kinetics because it is not hyperbolic. There appears to be a lag in activity at low substrate concentrations before the curve increases to give a sigmoidal shape. These enzymes are more sensitive to substrate concentration than non-allosteric enzymes. You can also see on this slide that there are two possibilities for effector types. One is a K system, shown on the left, in which the effector affects the Km but not the Vmax for the enzyme. The other is a V system, shown on the right. In this system, the effector affects Vmax but not Km. There are several different models to explain allosteric control. However, they all rely on conformational changes to change the activity of the enzyme. Here is a simple model which assumes that the enzyme has two domains, the regulatory domain having the allosteric binding site, the other with the active site. When the modulator binds to the allosteric site, it causes conformational changes in the regulatory domain that are felt at the active site, which may also undergo conformational change upon modulator binding. This long-range modulation of the active site provides variation in the degree of enzyme activity at the molecular level. If modulation makes the enzyme more active, then the substrate may bind more easily, in the case of the K system, or stabilize the transition state better, in the case of the V system. We would expect negative modulators to trigger conformational changes having the opposite effects. Among all the models to explain allostery in proteins, 3 have been particularly successful when applied to the allosteric regulation of enzymes. The Monod-Wieman-Changer model is the earliest and easiest to apply. This model considers oligomeric enzymes with identical subunits. The enzyme exists in 2 conformations with different activities, a relaxed form R, which is more active, and a taut form T, which is less active. Binding of an effector to the enzyme changes the conformation of all of the subunits simultaneously into either a more or less active conformation. Subsequent ligands can then bind more or less easily. This is a concerted model. The Koshland-Namathi-Filmer model differs from the MWC model only in 2 ways. Firstly, the enzyme only undergoes conformational change upon effector binding, and secondly, that a change in conformation of one subunit does not need to cause conformational change in the other subunits. Therefore, this is sometimes referred to as a sequential model. Mathematically, the MWC model is more easily considered and is therefore the one most often applied. However, not all the enzymes observed to have allosteric control are multi-subunit enzymes. Therefore, another approach is needed. In this approach, it is thought that the enzyme has a number of possible conformations that exist in a statistical ensemble that continuously change conformation in localized regions. These regions are most often disordered sections of the enzyme's structure. In this model, binding of the effector stabilizes some of the conformations over others. If the more stabilized conformations are more active, the effector is an activator, and if the stabilized conformations are less active, the effector is an inhibitor. Regardless of which of these models proves to be correct, they all rely on effector binding and conformational change. Next time, we'll discuss covalent modifications.